So I mentioned in the last video the sliding filament model of contraction. So what is that? Well, it's a contraction model that states that during contraction the thin filaments slide past the thick ones so the actin and myosin filaments overlap to a greater degree. So plain English, the sliding filament model of contraction says that actin and myosin overlap more when muscle contraction happens. Why is that important? Well, because you might think that actin and myosin shorten. That contraction means shortening, specifically of actin and myosin. That kind of sounds right. You're close, but not quite. Actin and myosin as myofilaments never shorten. Instead, they overlap more, which shortens the sarcomere. So when the nervous system stimulates muscle fibers, the myosin head latches on to the actin active sites, and sliding, or increased overlap, begins. This interaction between myosin and actin forms what we call a cross bridge, and these cross bridge attachments form and break several times during a contraction, creating propulsion forces that act like tiny ratchets to generate tension and shorten again the sarcomere. So only the sarcomere shortens, but it's because of increased overlap of actin and myosin. Actin and myosin never shorten. So this event inside of a skeletal muscle fiber would be occurring simultaneously in all sarcomeres throughout this cell during muscle contraction. So when a muscle cell shortens, the I-band shortens, the distance between the successive Z-discs that outline sarcomeres get closer, H-zones completely disappear, and the contiguous or adjacent A-bands are going to move closer together. Again, the length of the A-bands do not change, but they do move closer together. So what is that going to look like? Well, in a skeletal muscle fiber, that's going to look like a shortened sarcomere. And that happens in every sarcomere in a myofibril, so it looks like a shortened myofibril. So we're really starting to get into the physiology of skeletal muscle fibers. So really, let's go back to the beginning. Let's talk about what causes contraction. So let's start with the nerve stimulus for skeletal muscle. Skeletal muscles are stimulated by motor neurons of the somatic nervous system. The ends of these axons, called axon terminals, get very close to skeletal muscle cells. They don't quite touch, but they do form a junction between the motor neuron and the muscle fiber. And this kind of junction between the two is called a neuromuscular junction. It's made up of the axonal branches of the end of an axon for a motor neuron and a single muscle fiber. So when we're looking at the sliding filament model of contraction, we get the impulse for the contraction from the action potential that comes from the motor neuron. That's going to travel along the sarcolemma of the muscle fiber into the T-tubules and communicate with the sarcoplasmic reticulum to cause the release of calcium. And that's going to initiate that last step, that sliding filament model of contraction, where we define contraction only in terms of the activation of myosin and actin cross bridges. That's the force generating sites for skeletal muscle contraction. What we're doing when we're talking about the sliding filament model of contraction is really just explaining the relationship between the thick and thin filaments, or the myosin and actin filaments. And it's a cyclic process. It does begin with calcium release from the sarcoplasmic reticulum, and once that binds to troponin, it moves troponin out of the way, which pulls tropomyosin, and that is going to expose the actin active sites. Once the actin active sites are exposed, the myosin heads can bind, and they bind to the actin active sites. That forms a cross bridge, and that initiates contraction. The contraction is going to lead to increased overlap between actin and myosin, with myosin providing the propulsion thanks to the ATP that binds to the myosin globular head. So that's going to generate tension, and that series of steps just repeats. And all of that repeats because of the series of steps that began it all at the neuromuscular junction. The neuromuscular junction gets the action potential from the neuron to the muscle. And the next step is to get calcium release, and the calcium release leads to the contraction cycle that forms a cross bridge, and that's going to give us muscle fiber contraction and the buildup of tension. So now that we have kind of the basics in play, you can see that this doesn't necessarily cause shortening of the myofilaments, just shortening of the sarcomeres, and the shortening is what generates tension, And that tension is generated by cross bridges, which are activated once myosin and actin can communicate. And so the shortening happens in sarcomeres, never the myofilaments. All right, so now we've got, again, the basics in play, but you're probably still a little confused, and that's fair, because we kind of had to introduce some terms that we haven't quite discussed in depth yet, just so that you had this overview in your head. So let's take a step back and go to step 1 for how skeletal muscles contract. And we're going to start with the neuromuscular junction. Step 1 is activation at the neuromuscular junction, where the motor neuron of the somatic nervous system gets really close to a skeletal muscle cell or fiber. In order to activate the skeletal muscle fiber, we have to have an action potential travel from the neuron to the skeletal muscle fiber. Once it's in the skeletal muscle fiber, that creates a change in membrane potential across the sarcolemma, the entire plasma membrane for the skeletal muscle cell. That generation of an electrical current is what we need at the end of step 1, activation of the neuromuscular junction. Once you have that action potential, once you have that electrical current, you can move on to phase 2, which is excitation, contraction, coupling. This is going to link the electrical signal, the current that we just got called an action potential, to contraction. So the action potential is going to automatically propagate or move along the sarcolemma, and the sarcolemma is connected to T tubules. So that means that the action potential, the electrical current, can then travel inside the skeletal muscle cell. It'll travel inside via the T tubules, and the T tubules will communicate with the sarcoplasmic reticulum that hold calcium and lead to calcium release. And it's going to be that intracellular calcium ion level increase, or the increase of calcium in the sarcoplasm of the skeletal muscle cell, that's going to provide the final trigger for contraction. Because calcium can then travel to troponin and move the troponin-tropomyosin cascade out of the way, and that allows actin and myosin to bind and leads to phase 3, known as the cross-bridge cycle. All right, now we're going to do all of that again with more detail. All right, phase 1 happens at the neuromuscular junction. Phase 2 is going to be a connection between excitation and contraction. All right, so phase 1, the motor neuron stimulates the muscle fiber. This is activation at the neuromuscular junction. So first, an action potential, which can be abbreviated AP, arrives at the end of an axon called the axon terminal. These axon terminals are very close to the skeletal muscle fiber plasma membrane. At the axon terminals in the neuron, when the impulse, the action potential, arrives, it causes the release of a chemical called acetylcholine. Acetylcholine then travels across the space between the neuron and the skeletal muscle fiber plasma membrane and binds to receptors on that plasma membrane, or on the sarcolemma. Those receptors are acetylcholine receptors. Those receptors open once they bind to acetylcholine, and that changes the permeability of the plasma membrane, of the sarcolemma. That allows an influx of sodium, which is a positive ion, and that's going to change the charge across the plasma membrane of a muscle cell. This is actually going to move the charge inside of the cell away from a very negative number, which is called depolarization. And this is happening initially in just one location, so it's called local depolarization. And it happens at the area where the motor neuron has its axon terminals get close to the muscle cell sarcolemma. That's sometimes called the end plate. So this could also be called not just local depolarization, but end plate potential. That's going to be the ignition step in getting the action potential or the electrical current in the sarcolemma. All right, so let's summarize again. The action potential arrives at the axon terminal of the motor neuron. The axon terminal from the motor neuron releases acetylcholine, and the acetylcholine binds to receptors on the sarcolemma. Those receptors open, allowing ion permeability to change for the sarcolemma. We see an influx of positive sodium ions, moving the inside charge of the skeletal muscle fiber away from a very negative number. This is called depolarization, and is at first happening in one location, so it's called local depolarization or end plate potential. And that means we've succeeded in activating the skeletal muscle fiber at the neuromuscular junction because we have gotten an electrical current or action potential in the sarcolemma. So again, that's phase 1. All right, phase 2, excitation, contraction, coupling. So now we're going to take this action potential that has arrived in the sarcolemma and see it move. We're going to see it propagate. And the action potential is going to travel across the entire sarcolemma of the skeletal muscle fiber. And it's going to travel from the sarcolemma then to the T-tubules because the sarcolemma and T-tubules are connected. The T-tubules, as they carry this electrical impulse, are going to cause calcium release from the sarcoplasmic reticulum. Once calcium release occurs, that's the end of phase 2 because it's going to lead to the cross-bridge cycle, which begins with myosin binding to actin. And that starts phase 3, the contraction cycle. So let's take a closer look at the neuromuscular junction. Here you can see it's made up of 3 parts: an axon terminal, a synaptic cleft, and junctional folds of the end plate. When we're talking about a neuromuscular junction, we're talking about an axon ending that gives off several curly branches, and that collectively forms the neuromuscular junction, where these axon terminals get close to the plasma membrane of a skeletal muscle cell. You can see that in the image on the left. And even though it looks like the axon terminals get very close to or even touch the skeletal muscle fiber, they do not touch. Instead, if we look at the image on the right, we have one axon terminal which is filled with synaptic vesicles or cell bubbles that contain neurotransmitter. Then we have a space between the axon terminal and the skeletal muscle fiber sarcolemma. That's called the synaptic cleft. The space is the synaptic cleft. And then on the end plate, you would have multiple areas of the sarcolemma that have these folds called junctional folds. And the end plate would just be an area around all the multiple axon terminals that get really close to the skeletal muscle cell. And so at that neuromuscular junction, the axon endings release acetylcholine from their synaptic vesicles using exocytosis. The acetylcholine travels across the space, the synaptic cleft, and binds to acetylcholine receptors located in the junctional folds of the sarcolemma's end plate. So that's how we're getting types of signal from one cell to another. That type of signal being the action potential, which is the electrical current. Here is a kind of more in-depth image showing you the axon terminals and how close they get to the skeletal muscle fiber. If we look at the image on the bottom left, you can see that calcium influx actually causes the exocytosis of the acetylcholine from these synaptic vesicles. And it is in green dots and travels to the junctional folds of the sarcolemma, which is the dark red line. Again, the sarcolemma is the plasma membrane of a skeletal muscle cell. And then once acetylcholine in the right image in green binds to the purple acetylcholine receptor, it opens up channels that overwhelmingly allow a large amount of sodium to go inside of the skeletal muscle cell. And that's going to initiate the local depolarization, or kind of the spark we need as an electrical current in the sarcolemma. We do also break down acetylcholine because we want to be able to reuse it, so it's broken down by an enzyme called acetylcholine esterase. Again, acetylcholine esterase is the enzyme that breaks down acetylcholine, and some of that is actually reused to make more acetylcholine in the motor neuron. So step by step, what's happening in step 1, the neuromuscular junction is laid out on this slide. We get a nerve impulse called an action potential that reaches the end of the axon called the axon terminal. That initiates a voltage-regulated calcium channel to open, allowing calcium to enter the axon terminal. Calcium entry at the axon terminal is going to cause acetylcholine release by exocytosis from synaptic vesicles. The acetylcholine will then travel across the synaptic cleft and bind to specific acetylcholine receptors that are on the junctional folds of the sarcolemma. That's going to be when we have the action potential in the sarcolemma, because it initiates sodium influx or a local depolarization, and that's going to change membrane potential in the end plate on the sarcolemma. Alright, also you want to make sure that you watch this video. In your review PowerPoints, you're going to have access to this video, and it'll go through the events of the neuromuscular junction, which is part 1 for skeletal muscle contraction. If you're not sure where to find the links for this video, you can also Google A and P Flix, F-L-I-X, muscle contraction part 1, or neuromuscular junction, and it should show up on YouTube. If you can't find the video, you'll probably watch them in class, but if you're trying to find it on your own, this is also another good review image to look at first. It's showing you excitation at the neuromuscular junction. Step 1, electrical impulse called an action potential, reaches the axon terminal. That causes calcium channels to open, calcium goes in, acetylcholine goes out. Acetylcholine goes into the synaptic cleft and binds to the acetylcholine receptors, which then open. That allows for an influx of sodium, which is going to be when we have the action potential in the sarcolemma, because it causes a membrane potential shift or in-plate potential, also known as a local depolarization. And we don't want to forget step 6, which is when acetylcholine has to be broken down by acetylcholine esterase, because we don't want acetylcholine to remain bound to the receptors on the junctional folds of the sarcolemma, because that would mean the muscle cell would continue to be stimulated, but it also wouldn't reset and couldn't be reused. So we don't want to block new impulses from reaching the muscle cell. It would make it very difficult to control muscle activity. So the acetylcholine that's bound to the receptors is actually quickly destroyed, mainly by the enzyme acetylcholine esterase. And this occurs in the synaptic cleft of the neuromuscular junction. Alright, so that's a review of part 1 and part 2, and an in-depth review of the neuromuscular junction, which is step 1 of skeletal muscle contraction. Next, we're going to move on and look at excitation-contraction coupling, which is step 2 for skeletal muscle contraction.